All right, let's begin with glucose homeostasis then. What I'm not going to do is go into all the detail that the textbook does with regards to, kind of extra points and things and the diabetes part of it. I think you can do that. But what I will be doing is kind of taking a step back and looking at an overview of the topic, because what can happen is as we go through topics, we can look more and more at the details and become more obsessed with, do I know every single detail, and forget the larger context of where it fits in with the larger topic. And ultimately, the higher grades, the A's and B's, are given for this, or given to the students that can look at a question and see where this question fits into the bigger topic. Can they make a link between the different subtopics within the larger topic? Okay, so that's what we're going to be trying to do here. All right, so glucose homeostasis, let's start with the basics. Okay, so let's begin then. So when glucose levels rise, this is detected by the alpha cells, this is detected by the beta cells of the islets of Langerhans of the pancreas. Okay, beta cells of the islets of Langerhans detect this. Okay, so glucose levels rise. This is detected by the beta cells of the islets of Langerhans. They then release insulin. Okay, and the insulin is responded to by its target organs. So targets respond. Okay, and here we are talking about the liver, and we are talking about muscle cells, skeletal muscle, right? And they respond, and their response is accompanied by the lowering of blood glucose levels. Okay? Yeah, so glucose levels rise. This is detected by the beta cells. They release insulin as a response to that. Insulin moves around in the blood, comes in contact with many tissues, cells. However, only its specific target organs respond because only they have the right receptor, complementary to insulin. And those target tissues, organs, are liver and muscle, okay? And they respond by absorbing glucose from the blood, therefore lowering the blood glucose levels. Now, if the blood glucose, this process will continue to happen, and at some point the blood glucose levels will get too low. So in that case, it is detected by the alpha cells of the islets of Langerhans, just going to abbreviate that, of the pancreas. Now, yeah, one thing you should be careful of is not just to call them alpha cells and beta cells. I would always play it safe and just say beta cells of the islets of the islets of Langerhans in the pancreas. I know it sounds a bit wordy, but you know biology now, that's how it is. You've got to be specific. There might be more than one beta cell in the body. Many cell types might be called beta cells and alpha cells, so you don't want to get that confused. Okay, so the alpha cells of the islets of Langerhans detect this. They then release glucagon, which is another hormone, and this is then responded to. Target responds, and in this case the target is the liver. And what the liver does is it kind of breaks down its stored glycogen to glucose, release this into the blood, so the blood glucose levels rise. Okay, and there we have that. Okay, so if the blood glucose levels rise, we go through this, and if they fall, we go through this. Right? Now, I told you about the bigger picture here, and the bigger picture in this case is homeostasis. Right? How is this process monitoring, how is this process kind of monitoring the levels of glucose and then responding to it? That's what you need to be aware of. Okay, so think about it. We got an increase in glucose, that's our stimulus, right? We have our sensor, which is the beta cells or the alpha cells of the islets of Langerhans, of the pancreas. They release a hormone to make their effector do something. So our hormones are our communication pathway. So we got stimulus, sensor, communication pathway, which is the hormones, either insulin or glucagon, and they in turn have induced a response in their target organ, which is for insulin it's liver and muscle, and for glucagon it's the liver, but it makes those organs do something to reverse the original change. Okay, so in this case our effectors are the liver and the muscle, right? But in order to get our effectors to do anything, we've got to send them a message. And that message is sent in the form of hormones, the communication pathway. And what we needed to get the hormone released was to detect the original change in the first place. And that was done by the alpha and beta cells of the islets of Langerhans in the pancreas. Okay? So this is very important, that we never lose sight of the context of what we are looking at. This is one example of how homeostasis is achieved, right? So yes, you'll be revising how all this stuff happens, but you can't forget about the bigger picture, which is negative feedback. This is a mechanism of negative feedback, monitoring glucose levels, responding to them to reverse any changes to keep the level at where it should be. Okay? So I'll just make a point of that. This diagram could be getting a bit busy, but in any case, this is our stimulus. These are our sensors. Okay, we have our communication pathways, which is, in this case, it's the hormones, but in another system, it might be the nervous system, it might be a neuronal kind of action potential message that stimulates or takes information from a sensor to an effector. But in this case, it happens to be a hormone. Okay, so communication. Might need to change this one. Communication. And we have our effectors. Effector. And we have our response. Okay, which is, in this case, if the stimulus is rising blood glucose levels, the response is the lowering of the blood glucose levels. And if the stimulus is the lowering of blood glucose levels, then the response is the raising of blood glucose levels. Okay, so that's that. And now we'll look more closely at how this is happening, particularly the alpha and beta cells of the islets of Langerhans. How do they detect glucose and then secrete insulin or release insulin as a response to that? And secondly, how do our target cells respond to that insulin that was released by our beta cells? How do they respond to that? How does the insulin make them absorb more glucose? Okay, that'll be next. Okay, so let's look more closely now at the islets of Langerhans. Okay, so what I'll do here, so we're going to focus on this bit now. Right, so what's happening here? Now, the wider context is the structure of the islet of Langerhans. You'll need to know that. It's part of the pancreas. If we have the pancreas and we kind of cut across it and then turn it around, so maybe if we have our pancreas like so, kind of like a leaf type shaped organ, but then if we cut across it and look at a cross-section of that organ, what would we be looking at if we zoomed in? Now, say that's the cross-section. Now, within that cross-section, there'll be lots of cells, and most of those cells, so cross-section, let's say we take a small section of that cross-section, and this is what we're looking at here. Now, you'll have a lot of cells, okay, here. Now, around the outside, you've got your exocrine cells. So, most of the cells here are endocrine cells. So they're producing hormone, and it's exiting the cell and it's ending up in the space between the cells. Okay? But there are some cells which are arranged in a tube, cylindrical arrangement, and what they do is secrete enzymes directly into a hollow space, and that hollow space, or the tube that's formed by these cells, eventually joins up to form the pancreatic duct. Okay, so this is a cross section of the Islets of Langerhans. So these tubes kind of would pop out in 3 dimensions like this. Okay, and that, and so with this one, 3 dimensions. Okay, it would kind of come out towards you. But then, so what these cells are doing, the cells that make up this tube, they're producing enzymes. And this is one of the functions of the pancreas, which is to produce enzymes which are used in the digestive process. It's a separate function from the production of hormones, but you do need to be aware of it. So these tubes eventually join up to form the bile duct. So they would join up like so. And in 3 dimensions, it would look something like this. But all the cells that form these tubes are releasing enzymes, and so the enzymes kind of collect in this space here, and they eventually join up with the digestive system at the duodenum. And so as your food comes past the stomach and enters the duodenum, it mixes with these enzymes, and these enzymes get to work on the proteins, on the lipids, on the carbohydrates, and by hydrolytic reactions break them down into their constituent components, amino acids, fatty acids, and monosaccharides and disaccharides. Okay, so that's that, right? That's the exocrine function of the pancreas. But as well as these tubular cells, arrangements of cells that release enzymes, we also have other cells, and it's here we have our alpha and beta cells kind of dotted around. Okay, so I'll put an alpha sign on some of them and a beta sign on other ones. Okay, but these are our alpha cells and beta cells in the islet of Langerhans right there. And they are producing either insulin or glucagon in response to the blood glucose levels. How do they know? How do they get exposed to the blood glucose levels? Well, we have blood vessels, capillaries, that kind of go through. And again, I'll show you in 3 dimensions. If we cut a section across, the tube would be coming out if we could see it. So this blood vessel, if it was going to carry on, would look like that. Okay, it would be coming out towards us, like so. Okay. Anyway, so the blood is moving through. Just like all organs have an artery which splits up into capillaries, and the capillaries then go through the organ, through the cells, rejoin to form the vein that goes back to the heart. In the same way, you've got an artery which goes, towards the pancreas. It splits up into these capillaries. These capillaries, they go through the pancreas in this way, and then eventually they'll reform to form the vein and take the blood away back to the heart. But because these capillaries go right through the pancreas and go through the islet of Langerhans, what it means is that the blood is moving right close to all these cells, the alpha cells and the beta cells. So if you've just had a meal and the digestive enzymes have broken down your chips or whatever into glucose, and that glucose has been absorbed into the blood at the intestines and then is now circulating, and so as the blood then moves through the Islet of Langerhans, some of that glucose comes out and it kind of surrounds the alpha cell or the beta cell. And in that way, the alpha cell or beta cell gets exposed suddenly to this high or low level of glucose, okay? Depending on if you've just had a meal or not. Okay? So, alpha cell right there. And, so you got alpha cells, and as the blood moves past, some of the glucose comes out, and you got the glucose will then be surrounding the beta cell, and then we'll look later at what effect that will have on the beta cell. Similarly, if it's been a long time since you had a meal and your blood glucose levels are low, this is then sensed by the alpha cell. So as the blood moves through the Islet of Langerhans, it moves past the alpha cell, it will be sensing that there are low levels, so there will be very little glucose surrounding that alpha cell, and it will then respond to that. Okay? So the blood, the capillaries that go through the Islet of Langerhans is very important because it exposes the alpha and beta cells to the blood glucose levels, and they can then make a response to that. Similarly, when they release their hormone, so if in the case of the beta cell, if there's high levels of glucose in the blood that's moving past the capillary, moving through the capillary, the beta cell is going to be producing lots of insulin. That insulin is going to build up around that cell and eventually diffuse into the capillary. And once it diffuses into the capillary, it becomes part of the blood circulation, and that it will eventually go around the whole body. Okay? And it will be diffusing into the capillary because, remember, it's going to have a high concentration around the beta cell because the beta cell is producing it. It's going to have a low concentration in the capillary because the capillary is not producing it. So it's going to move in there. And it will, wherever it goes, it's going to be diffusing out of the capillary because nowhere else is producing insulin. So it's got a low concentration everywhere else, so it diffuses out of the capillary to all the tissues, but only those tissues with the right receptor to respond to insulin will it have any effect on. Is that making sense? Okay, so that's the kind of general arrangement of the islets of Langerhans, and now we'll focus more closely on what is happening within one of these beta cells when the glucose level goes up. Okay guys, so let's continue. I've just kind of prepared this not to waste time. I like to keep my videos short and sweet. So, here we go then. So what we're looking at here is a beta cell of the Islet of Langerhans in the pancreas. Okay, so just one of these cells. And remember, our situation was, we've just had a meal. Thanks to the digestive enzymes released by the pancreas via its exocrine function, you just had chips. The starch in the chips have been broken down by the amylase and the maltase in the pancreatic juice released by the pancreas. So those enzymes and the chips have mixed together. The starch has been broken down thanks to those enzymes, ultimately into glucose. The glucose is small, can be absorbed into the blood. The blood is moving around. The blood arrives it arrives at the Islet of Langerhans in the pancreas via the capillary. That high glucose causes the glucose then to diffuse out of the blood capillary amongst the cells of the islets of Langerhans in the pancreas, and that high glucose levels is now surrounding this cell. Now, how does that lead to this beta cell releasing insulin? That's the question. Because remember, we've just had high glucose. How do the beta cells detect that and release insulin? This is what we are looking at. So glucose is surrounding this cell, right, after a meal. Now this is a beta cell, and there are a number of components that we need to be aware of. First, there's a glucose transporter in the cell surface membrane, which allows glucose to move down its concentration gradient, allow it to diffuse from the outside to the inside. Now it needs a special protein for this to happen because glucose is polar. It cannot dissolve in the membrane. So, because the membrane, remember, is hydrophobic, so glucose is polar, hydrophilic, so it needs a protein to allow it to pass that hydrophobic barrier. Anyway, the channel is there, and it will allow glucose to move according to its concentration gradient. If it happens to be higher here, it will go out. All right. Next, we've got potassium ion channel in the membrane, which allows the movement of potassium ions down its concentration gradient. It will be very similar to the ones found on neurons. Okay. Our third character is the calcium ion channel. It allows calcium ions to move down their concentration gradient. But this one is voltage gated. That's important, but we'll see later. Okay? These are our main players. Also, insulin is produced inside this cell. Shouldn't be a surprise. If beta cells produce a hormone, it doesn't magic out of it nowhere, right? We've got a nucleus, right? It encodes enzymes that produce insulin, you know. We've got DNA in here. It's probably got a gene to string a few amino acids together, right? So those amino acids must be put together on ribosomes, endoplasmic reticulum, Golgi body is probably involved to produce ultimately the string of amino acids called insulin. And they end up in these vesicles thanks to it being synthesized via the endoplasmic reticulum, Golgi body, packaged into vesicles. Bingo, we've got vesicles that have insulin, but they're inside the cell. Okay, membrane, insulin inside. Okay, they're inside the cell. How do they come out? So, high glucose outside the cell, glucose then diffuses in via the glucose transporter. So we've begun our sequence of events. Okay, glucose diffuses in because there's loads of glucose outside thanks to our capillary, just had a meal. So glucose then diffuses in, and now we have glucose within the cell. Okay. Now glucose enters the cell, but what happens next is an important enzyme called glucokinase adds a phosphate group to the glucose, converting it to glucose 6-phosphate. Okay, so it adds a phosphate group to all of these glucose molecules coming in, glucose 6-phosphate. Now this glucose 6-phosphate can't go back out. Okay, it's locked in. And also, once you phosphorylate glucose and add a phosphate group to it, converting it to glucose 6-phosphate, it is then committed to entering the respiratory pathway, okay, the aerobic respiration pathway via the mitochondria. So it will then enter that pathway thanks to glucokinase phosphorylating the glucose, and what we get out of it, of course, as well as carbon dioxide and water, but they're not important to us right now, is ATP. So, loads of glucose outside, lots of glucose inside. Lots of glucose inside, lots of glucose 6-phosphate thanks to glucokinase. Lots of glucose 6-phosphate means we have lots of ATP production. Hope that's making sense. Now, this is the bit that you'll have to just remember. ATP causes the potassium channel to close. Okay, now remember, well, not remember, but this cell has a kind of resting potential of approximately minus 70. I've seen minus 65 quoted. Potassium ions have a tendency to move out of the cell. These channels are open, and to maintain the normal resting potential of minus 70 millivolts, there is a permeability of this membrane to potassium ions, and they tend to go out of the cell. Okay? However, when there's lots of ATP, this causes the potassium channel to close. Okay, so the potassium channel closes, and because we're retaining more positive ions in the cell, we start to get depolarization. So the inside of the cell, instead of being negative in relation to the outside, starts to become more positive. Okay, so we start to get a depolarization because of this. Okay, because it used to be very negative inside here, but because we're stopping the potassium ions from going out, they're being kept inside, and the inside is now becoming more positive, so we're getting a depolarization, and this depolarization affects the voltage-gated calcium channels. Okay? It affects the voltage-gated calcium channel in that it causes those channels to open. Okay? So, and these are, this should be a very kind of similar idea to what you did in the synaptic transmission. Similar ideas. Depolarization causes voltage-gated calcium channels to open. Calcium enters the cell, and similar to what happened in the synapse, calcium entry into this cell causes those vesicles that contain insulin to fuse with the membrane, to move towards the membrane and fuse with it. And when the membrane of the vesicle becomes one with the membrane of the cell, and please don't use that language in an exam, but when they become essentially one membrane, that's the fusion process, the contents of the vesicle technically become outside the cell. Okay? So this insulin is now free to diffuse away from the area of its high concentration, out, away from where it was. Okay? So that process is exocytosis. Okay? Movement of the vesicle towards the membrane, fusion of the membrane of the vesicle with the cell membrane, and the release of the contents. Okay? So this insulin is now outside. Alright? And that's that. Okay? So that sequence of events took us from having a meal, high levels of glucose in the capillary, glucose diffuses out, surrounds the beta cell. Glucose enters the cell thanks to the glucose transporter, gets phosphorylated, and in doing so generates lots of ATP via aerobic respiration. Lots of ATP is then produced, blocks the potassium or causes the potassium channels to close. They close, potassium remains in the cell, causes a depolarization. The depolarization affects the voltage-gated calcium channel. Calcium channels open, calcium enters the cell. Calcium entering the cell causes the insulin-containing vesicles to fuse with the membrane and release their insulin into the extracellular space. And once in the extracellular space, it will then make its way into the capillary, because the capillary has a low concentration of insulin. Insulin goes into the capillary and off it goes around the whole body, diffusing out of the capillary in far distant tissues. And if those tissues and cells have the right receptor for insulin, those cells will respond to the message released by the beta cell. Okay, so that is the beta cell. It looks a little bit confusing, but if you might just need to rewind and just watch that again. It's the sequence of events that's important, not the absolute information that you see here. Okay, what we'll do next is look at then how distant cells will be responding to the insulin that's now moving around in the blood circulation. Okay, so we'll look at that next, and we'll take the example of the liver cell. All right, so what we've done is we've looked at what happens. The glucose levels have risen, Beta cells have detected it, they've released insulin as a result. Now, as we speak, the insulin is circulating in the blood, and we're going to look at what happens when it diffuses out of a capillary in the liver. Okay, how do the liver cells then respond to that insulin? How does that bring about glucose levels going down in the blood? So what happens next? All right, so where did we leave the story? Insulin has been released. What's probably going to then happen is it's going to diffuse into a capillary, and that capillary will then transport that insulin around the body. Okay, now let's say the insulin travels around the body, around the circulation, and then it arrives at one of our target organs. Now in this case it's the liver. What happens? So remember, this blood contains loads of glucose, okay, which we need to do something about, and it contains now lots of insulin too. Okay, right. So what happens? Obviously high concentrations, both will diffuse out. So we get both diffusing out of the gaps. We get lots of insulin out here, and we get lots of glucose. Okay, glucose. All right. Okay, so I'll just we'll just call this what it is: the hepatocyte. Hepato meaning of the liver. Okay. Now, so right, well, lots of insulin. Now, what happens? So remember what we said. Every hormone needs a complementary receptor for that hormone to have an effect on a cell. Now, insulin has a complementary receptor, and that receptor will be on the surface of all the cells that can respond to insulin. And the liver cell, the hepatocyte, is no different. So this insulin binds to its complementary receptor. Okay, because it has a complementary shape, it binds a receptor. It's polar, insulin is polar, and so it can't cross the membrane, so it has to bind the cell surface receptor on the outside of the cell, and the message is transduced via this protein which spans the membrane. Now, when the insulin locks into its complementary receptor, it activates an enzyme called adenylate cyclase. This enzyme converts ATP into cyclic AMP. Okay, and cyclic AMP is essentially it's the message, it's the secondary messenger, and we say that because, well, we covered that in the previous video, didn't we? Yeah. So insulin, the hormone, binds outside. It causes a production of something inside the cell, which then causes various things to happen inside the cell. So cyclic AMP initiates many signaling pathways, all right? So it makes the cell do lots of things. Various enzymes are activated that makes the cell carry out new processes, and those new processes make the whole cell behave differently simply because the hormone bound the cell. But that's how the hormone makes the effector do new things, because it's making all these new signaling pathways happen inside the cell. Okay, so loads of glucose outside, it can't go anywhere yet. The insulin is bound, and cyclic AMP is produced. Many signaling pathways ensue, and one of these signaling pathways causes vesicles that are contained in the hepatocyte, similar to those vesicles, but they don't have anything really of significance inside them, to my knowledge. What's more important is this protein that's embedded within the membrane. Again, thanks to the endoplasmic reticulum, ribosomes, Golgi apparatus for getting all this stuff ready. Okay? Now, this protein on the membrane of the vesicle is a glucose transporter protein. Okay? Now, when cyclic AMP is produced, signaling pathways are initiated. It causes these vesicles to fuse with the membrane. Again, a recurring theme, makes revision easier for you. Anyway, when this vesicle fuses with the membrane, its membrane becomes one with the membrane of the cell, in a way. And so the cell now has, as part of its membrane, it has these glucose transporters on its surface, thanks to the process of insulin signaling. So because that vesicle with the glucose transporter has fused with the membrane, the glucose transporters are now part of the cell membrane of the hepatocyte, and all this glucose that's just waiting outside with no way to get in, now it's got a way to get in, because these glucose transporters allow glucose to enter the cell by diffusion down its concentration gradient. Okay? And thus all this glucose will now rush into the cell by diffusion. Okay? Now, that can't be the end of the story, because eventually we're going to reach a point where there's so much glucose inside that no more can come in. Remember, these processes are happening by diffusion. The movement of glucose is happening by diffusion. If there's a high concentration of glucose within this cell, more will not enter, and therefore the liver will not be able to carry out its function of lowering the blood glucose levels. So glucose enters, and it must be utilized. It must be used up in some way that we maintain a low concentration of glucose inside this cell. So, just going to make a little bit of space here, but there are a number of ways that you can use glucose up. And I think it's important to just state that these processes are also stimulated by the cyclic AMP. So one of the things that the cyclic AMP did was induce vesicle fusion with the membrane, but the other things it's going to do is activate enzymes that convert glucose into glycogen. So they polymerize the glucose, remember AS biology, glucose to glycogen, carbohydrate, polysaccharide, condensation reactions. Okay, so it's going to activate those enzymes required for that, and that process is called glycogenesis. Okay, creation of glycogen. All right. So some of the glucose will be converted into the insoluble glycogen. Some of it will be used to make fats in a process called lipogenesis. Okay, right? I think I'm missing something out. But anyway, these are oh yes, and also respiration. Okay, glucose can also be used up in respiration. So these three processes ensure that the glucose concentrations are not allowed to build up in the cell. We are converting it into glycogen as a store, right? So it's not technically going anywhere, but we're converting it to glycogen so that more glucose will be able to come in. Okay, and then later when we need glycogen, it can be broken down to release the glucose again. Okay, so yeah, I think that's it, right? So in this way, if we maintain low concentration of glucose within this hepatocyte by these processes, converting glucose to glycogen, using glucose to make fats, but breaking down glucose in respiration, glucose will be able to continue to move in down its concentration gradient through the glucose transporter, and we will slowly, well, maybe if many cells are doing all this at the same time, it's going to very quickly reduce the blood glucose levels. Okay, right? And as the blood glucose levels go down thanks to this process, over here there's less of all this going on because we've got less glucose going around in our capillary. So less glucose going around in our capillary thanks to the liver, less glucose coming in, less glucose being phosphorylated here, less ATP made, potassium ions are therefore open, we don't get depolarization, we don't get calcium entry, and we don't get release of insulin. Yeah, kind of magic. Yeah, and that, I believe, is that. I'm not going to go through the process of glucagon. It's pretty much, similar kind of parallels, So I'm not going to go through that again, but I hope that that has given you a bit of context. You know, this you're going to do, it's going to take a lot of revision to kind of get these things down. All right? But don't forget where these fit into the bigger picture. Okay? Right? This has to happen so that this can happen. Okay? All right, guys. Good luck.